Swayam Prabha. Digital India. Educated India.
Hello, my dear students. Welcome to Swayam Prabha, sponsored by MHRD, New Delhi. Course title Medicinal Chemistry. Lecture title Introduction to Combinatorial Synthesis. Faculty name Tamak Vikrant Murlidhar. Module number 7, lecture number 11. The contents of this session: Introduction to Combinatorial Chemistry, types of combinatorial synthesis, and these are the types of combinatorial synthesis. First one is parallel synthesis, second one is solid phase synthesis, and third one is mix and split method. And again, this parallel synthesis, it is having subtypes, and after that we will be looking examples of combinatorial synthesis and its limitation. So advantages of combinatorial synthesis we will see in introduction part. So let's begin with content of this lecture. First, content. So what is actually combinatorial chemistry or combinatorial synthesis? Combinatorial chemistry comprises chemical synthetic methods that make you possible to prepare large number of compounds in a single process. So that large number of compound, it may be 10 to 1000 or even millions. And whenever we go through a traditional synthesis method, a chemist or medicinal chemist will be able to only synthesize a specific chemical or a single moiety. But by use of this technique of a combinatorial chemistry, a chemist can synthesize lot many compounds at a time in a single process, and this process is said to be a combinatorial synthesis. And all this, it is possible in single reaction vessel. This technique uses same reaction condition with same reaction vessel to produce large number of analogues. So how actually it happens? So we'll take an example. See, here you can see a green color moiety, or we can say a green color molecule, and this green color molecule, it is reacted with yellow color molecule. Now again, at the same time, it has been reacted with an blue color, and after that a red color. So now we will be getting three products, and these three products, now they are combined, and as three products are being synthesized, from these three products we can synthesize more libraries. So now we can introduce, as we have got all these three products, and from these three we can add again yellow, again blue, and again red. So there will be one more linkage of a compound. Firstly it was just a single, now it has become a triplicate, in between duplicate and now triplicate. So now there are three sequences of a compound coupling, and here you can see almost 9 compounds they are been formed from only 3 compounds. So by this way, the combinatorial synthesis works, or combinatorial chemistry works within single reaction vessel. These reactions can be carried out, and now these are the products which are available. So similar way, lot many products can be synthesized at the same time, but there are some limitations, like the reaction conditions are same. So it may happen that some reactions will work or some they will not work. And most of the time when combinatorial chemistry it has been done of any compound, so reaction conditions are taken such that they are suited for that reagents and reactants. And one more disadvantage of this combinatorial chemistry is that once these compounds they are been formed in a single vessel, so it is difficult to isolate, or it requires a time to isolate. We will come across and what are the limitations of an combinatorial chemistry at the end of this session. So from this figure it has been understood that what is actually an combinatorial chemistry or combinatorial synthesis. So we'll see firstly an advantages. As we have seen how combinatorial synthesis works or combinatorial chemistry works. So from that it can be concluded that it is fast method. As the medicinal chemist, if he starts with a conventional synthesis, he can only synthesize only one product at a time. But by use of this combinatorial method, the process can be very fast. Combinatorial approach can give rise to a million of compound at the same time, as well as it will take to produce one compound by traditional method of synthesis. After that, economical. A negative result of mixture saves the effort of synthesis, purification and identification of each compound. So here it means that whenever the product it is been synthesized by an combinatorial chemistry, and if certain groups it gives a negative result. So negative result in sense the pharmacological activity. When there is HTS, that is high throughput screening, in that case many compounds are being synthesized by an combinatorial chemistry, and that particular vessel containing in that compound they are directly been acid by an pharmacological technique. And if it gives a negative result, then there is no need of an purification and identification of that compounds, because once that vessel it is negative, so chemist goes for an next type of an vessel or next type of an compounds which are been prepared by this combinatorial chemistry, and they find an assay, and the assay which gives a result, positive result, that assay only it is been purified, or that compounds are only been purified and identified. So that is why it is an economical, because whenever we go by a single synthesis method, so it will take huge amount of time as well as to predict each compound's pharmacological assay. So it requires a cost. So that is why this method is an economical. After that, easy isolation, purification and identification of active molecules from combinatorial lab is relatively easy. So this is because we know what type of compound is been kept in what type of a reaction mixture. So we will come across this thing why it is said to be an easy, because in parallel synthesis or solid phase synthesis we will come across it. After that, drug discovery. So mixed combinatorial synthesis produce a chemical pool. It is one of a method, that is split and mix combinatorial synthesis method. Probability of finding the molecules in random screening process is proportional to number of molecules subjected to the screening process. At same time, lot much library it has been synthesized. So potentially we may found lot many compounds or a series of compound which are showing an positive results at the time of an screening. After that drug optimization, parallel synthesis produce analogs with slight difference which is required for lead optimization. So in the last lecture we have seen how lead it is been optimized, and at the time of an lead optimization, so there is slight difference in an analog, and we have also seen what is an analog in the previous sessions. What is analog design? So analog design, it is little bit just an different design or different or introduction of some little molecules in total drug, and little bit modification, it improves pharmacokinetic and pharmacodynamic properties of a drug which are required to be in therapeutic, showing a therapeutic value of a drug, and it helps in an lead optimization also. So these are some advantages of an combinatorial chemistry. Now we will discuss next point, that is types of combinatorial synthesis. So first method, that is parallel synthesis method, and this parallel synthesis method name itself indicate that the reactions are carried out in the parallel way. So there are three methods in parallel synthesis. One is T-bag synthesis, second one is multi-pin method, and third one, that is spot membrane method. So in case of an T-bag, so it is indicating that T-bag means it is a small bag, and this small bag it is having a resin. After that, multi-pin method, so pin which are multiple in number, having some chemical moieties attached with it. And after that, spot membrane method. So spotting on a membrane, it has been done in case of an spot membrane method. So we will elaborate each method. After that, second class of this synthesis is solid phase synthesis. So again, name itself indicates solid. So there are some anchors and linkers which are joined to an solid resin, and reaction it has been carried out. After that, mix and split method. So mixing and splitting, these are done simultaneously in this method, and after mixing, again splitting it has been done, again mixing it has been done. So it continues till our library it has been developed. So these are the three methods of combinatorial synthesis: parallel synthesis method, solid phase synthesis method, mix and split method. So we'll see one by one. First one, that is parallel synthesis method. So in case of an parallel synthesis method by which library construction occurs in distinct spaces on an array, such that each member of an library is defined by its position on that array. So we can understand this statement with a figure. An inherent property of this methodology is that the structure of library member can be deduced from its position on the array. So both this concept, it indicate that the structures can be easily identified by this method. So how it is, we will see it one by one. See, here you can see A is one of a chemical, and this A chemical, it is linked with different chemicals. So this statement says that whenever there is construction of a library, so each member of a library is defined position. So all the positions are been defined. That is A, it has been reacted with all the compounds, And also it is easy to identify the structure of a compound what has been prepared, or it is easy to deduce the structure of a compound what has been prepared from the position of the position on the array. So it is because we know the structure of A and we also know the structure of B. Once it combine, we can easily identify what kind of a structure it might be. Or if you take an example that structure A is known to us and structure F it is known to us, and once it combine, easily chemist can identify what kind of a structure it has been prepared or been synthesized. And from the position, because all this they are been labeled, and from these positions one can easily identify what kind of a compound it is or what kind of a structure that molecule is been having. So this is first method of an parallel synthesis. So it is said to be an T-bag synthesis method because a material which is been utilized, so it looks like an T-bag, so it is said to be an T-bag synthesis method. This method was developed in 1985 by Hongton for multiple peptide synthesis, and sometime it is said to be an Hongton T-bag synthesis. So he was a scientist who prepared an peptide by this ways in 1985. Braid of solid support are enclosed with permeable polypropylene plastic bags having a size 5 into 2 mm mesh packets, then placed for coupling into a reaction vessel containing a solution of amino acid and coupling reagent. So bags, they are like an tea bag, and these are having a particular size, and they are also having an permeable polypropylene which allows an reagent or soluble solutions to enter, but they does not allow resins and linkers to come out, because resins and anchors they are not soluble with the fluid whatever we are using, and this polypropylene does not allow to come out that material. And these amino acids and coupling reagents, they are soluble in the fluid whatever we are taking, so it may be water or it may be any organic solvent, and due to which there is coupling, and because reaction conditions are been maintained, a tea bag mesh size is too small to allow the resin beds to escape. So that is what we were discussing, that resin beds are not easy to escape, or they will not escape from an tea bag, because the mesh size is important. It is having very low mesh size, but it allows the soluble reagents to enter, and it is because they are soluble, and that is why they can be entering into an bag. Such kind of bag it is having 22 mesh size, and the size of this it is 5 mm, and so this thing is, it is nothing but a resin. Resin, it is having an anchor and a polypropylene mesh. So polypropylene mesh, it is permeable only for solvents and soluble reagents to enter. So so that is why it is said to be a tea bag, because it looks a small tea bag, and this resin it is having an anchor with it for coupling, and the soluble reagents or the amino acids from which we need to prepare a peptide, so they are been solubilized within solvent, and this tea bag it is been kept within solvent. So this is how it works, a polypropylene bottle, and this polypropylene bottle it is full or filled with an solution of an amino acids and coupling reagents, and once it has been filled with an amino acids and coupling reagent, so this polypropylene mesh, polypropylene mesh, it allows only entrance of an solution into this bags, and one solution it enters into the bag, so there will be coupling, and due to this coupling it will form an peptide bonds, and this peptide ultimately it results into a protein. So now we'll see the process of T bag synthesis. So T bag, they are being labeled, and this is an advantage of this method, that easily we can identify what type of an peptide it has been formed, because we know what type of an compound T bag it is having inside with an resin linked, and after that we also know what kind of a solution of an amino acid it has been present in the bag, or it is present in the bottle, and what kind of a linkage it will form. So already it has been predetermined, and it is easy to identify because these tea bags are been labeled. So first step, that is cleavage. So the cleavage here, it means that a tea bag, it is been protected, or the resins which are linked with an anchor or linker, they are been protected, and that is why there is need to cleave the end atoms, and for this purpose piperidine or DMF it is been utilized. After that, washing step. So in the washing step, solvents such as DMF and methanol, they are being utilized. So to remove, once it has been cleaved, so it has been protected, and this protected group, it has been, once it has been cleaved, so it has been washed, because that protected group which has been cleaved in the first step, it gets solubilized into DMF and methanol. Once it is ready, after cleavage, now it has been activated, and once it has been activated, so now here you can see solutions of an different amino acid, they are been introduced, and as these bags are been labeled, so we know what kind of an linkers they are having and what kind of an peptides they will form. So already it has been known by a medicinal chemist. So here some of an amino acids such as alanine, isoleucine, histidine, tyrosine and lysine, they are kept for coupling, and so this step, it is said to be an coupling step. After that, washing step. So washing step here, it means that excess of this amino acids which are been present, they are been solubilized with the DMF and methanol, and once it has been solubilized with the DMF and methanol, now compounds with the tea bag, they are ready. Second method of parallel synthesis, it is multi-pin method. This method was published by Geisen and his colleagues in 1984. The multi-pin apparatus has block of wells. So there are total 96 wells in a format of 12 rows and 8 columns, serving as a reaction vessel, and cover plate with mounted polyethylene rods or pins fitting into well. The end of polyethylene rods or pins are coated with deodorized polyacrylic acid. The protected amino acid used in building the peptides and the coupling reagents are dissolved and added to the walls. The coated end of pin are immersed into a solution and kept until the coupling reaction ends. The peptide formed on the pins are immersed into solution. So this is how actually and vertically it looks. So these pins, they are been coated with an derivative as polyacrylic acid and protected amino acids which are dissolved into the wells, and the coated ends of in this, here you can see a black ends, they are been kept or dipped into a solution until the reaction it ends, and now the peptides are formed on the pins, and here the compound of desired it will be forming. By looking overway, we can see such kind of an wells. So there are 12 wells, as it was been said earlier, into 8 rows, and total 96 reactions can be carried out in a single at a single moment. In conventional method, a reaction or chemist can synthesize only one compound at the same time, but with the use of this multi-pin method, so 96 reactions can be carried out at the same times, and the product it is not in the well, so it is with the pin, and this pin it will be having a desired peptide which has been formed. So it is very convenient way to synthesize compounds on a large scale, on number of scales, because 96 compounds they are being formed at a time. But some disadvantage of this methods are also that reaction conditions are same, and due to reaction conditions are same, at some instance or in some wells the compounds cannot be formed because they require more drastic conditions, or the conditions whichever are being kept are not sufficient to carry out that reaction. So all the time it will not happen that 96 of 96 peptides are being formed. So there might be there will be and some limitation that some of the peptides they will remain unchanged, or there will be no reaction, as conditions should be little bit drastic for it. And also one of the disadvantage of this method is that, so keeping this 96 wells at a time and maintaining the temperature for each well, it does not happen, because in case of a conventional method, one can, with the use of a round bottom flask, a chemist can optimize the temperature or it can maintain the temperature. But whenever this type of a method it is been utilized, so it becomes very difficult to maintain the temperature for all these 96 wells. And one of an advantage of this method is that because we are knowing what kind of an chemical does each well has, and it has been labeled, so what kind of an label, what kind of an chemical it has been having, so it becomes easy, because a pin it is having a particular reagent that is derivative as polyacrylic acid, and what type of an chemical will couple, or what type of an amino acid it will couple with an pin, so already the chemist knows it because labeling of in these wells. So it is easy to identify and structures also, or it is easy to deduce the structures also. But some more tedious jobs are that to isolate each and every structure, it becomes a little bit difficult. Next method, that is spot membrane method. So name itself indicate that there is membrane in this method, and spot, so defining a spot or putting a spot on a membrane. This is how the parallel synthesis works for a spot membrane method. So we'll see in the detail. So it was been introduced by Frank in 1992 and his group, also for preparing peptide arrays. So all these three methods of parallel synthesis, they have been developed after 1980s almost. The synthesis carried out on cellulose paper membrane instead of polyethylene pins as solid support for peptide synthesis. Small droplets of solution of protected amino acid dissolve in low volatility solvents and coupling reagents are pipetted on predefined position of the membrane. A spot thus formed can be considered as a reactor when the conversion of reactions of solid phase synthesis takes place. So this is how the cellulose paper it is. So instead of using polyethylene pins in case of an earlier methods of an parallel synthesis, so cellulose paper membrane it has been utilized, and the small droplets of solutions of protected amino acid dissolved in low volatility solvents are been utilized. Here low volatile solvent is utilized because whenever we are using an high volatile solvent, so it will be easy to solvent for an evaporate, and there will be there may be a chance of no reactions. For that purpose we need an compounds with an high boiling point so that it will not evaporate, because there will be no medium for an reactions once a compound or once a solvent it has been present on it. And coupling reagents which are been coupled, which we have to couple, so they are pipetted. So by using such kind of an pipette, automated pipette, so we can spot on each, and due to which it is having an advantage that what kind of an small droplets of an protected amino acids we have spotted over here, and what we are spotting by using a coupling reagent, so we know it already, and due to which it is easy to identify and compounds by this method also. So these are the three methods for an parallel synthesis. So till yet we have studied it. Next method, that is solid phase synthesis. So name itself indicates that it is having a solid, and this solid is having a resin, and resin it is linked with an anchor or linker, and after that it will couple with certain reagents to form an peptides. Solid phase synthesis is a method in which molecules are covalently bound on solid support material and synthesized step by step in single reaction vessel utilizing selective protecting group chemistry. So in case of this solid phase synthesis, molecules are having an covalent bonding, not an ionic bonding, on solid support, and step by step this reaction it is been carried out in a single reaction vessel. And also some protecting groups, so we will come across what are the type of protecting groups in chemistry, like BOC or tertiary butyl. So these are some protecting groups which are being utilized, so there will be no reaction at that active site. So actually how this solid phase synthesis it is, so we'll see by this figure. So here you can see a resin having an X group, so that is nothing but a linker, and this linker now so this amino acid which is present over here, so it has been loaded into a vessel, because this reaction it is carried out in a single vessel, but it is step by step. So from this figure also we can know that how it has been carried out by step. After that, washing, deprotection, and again washing it has been done. So this all the process they are been done to activate this amino group or to remove protected amino acid group. So NH it was even protected by R, and we need to remove that R. So firstly it has been washed. So washing it is done to remove excess of an resin or amino acid, and because in washing, so this compound or whatever complex it has been formed with the resin and amino acid, so it will be not soluble. and after that it is been deprotected. So deprotection, it means that activation of an NH2 from an secondary structure. Now it is an primary structure, and after that again washing it has been done. So washing it has been done to remove this R group, and in case of an washing, so solvent which will be utilized, so this complex, it will be not soluble with that solvent, only the R group it will be soluble with that solvent. So it has been now removed. Again one more reagent it has been added, and now it is ready for coupling, because here you can see that NH2 it couples with carboxylic acid and forming an amide in between, and now this is nothing but an peptide bond. And this is how the peptides are been synthesized by this method. And again process it has been repeated, that R it has been washed, or by washing this complex molecule R will be removed, and R as soon as it has been removed, so there will be activation of an NH2, or there will be, and again NH over here, and again one more reagent it will be coupled, and coupling of this it continues until the desired library it has been formed. So this process it continues until the desired library it has been formed, and once a desired library or desired peptide it has been formed, so after that our compound it is almost ready. After that there is cleavage from a resin, because this one is not a desired one, and this carbonyl terminal it has been converted to an carboxylic acid, and now our protein molecule it is now ready. So this is how proteins are being synthesized by solid phase synthesis, and this method it is done in single vessel. So from all this process one can understand that this method it is done in a single vessel, and it is done by step. So from this process it has been understood that it is done step by step. So the role of resin and linker, it is just only to form an peptide and form a chain, and after chain it has been form of an peptide or desired protein it has been formed, so it has been cleaved, and as soon as it has been cleaved, now desired product it is ready. So now we'll see what are the requirements of a solid phase synthesis. A resin bed or functionalized surface act as solid surface, it is required. So resin beds, as name itself indicate that they are solid in nature, and it is required for a coupling of an anchor or linker. And once anchor or linker it has been coupled, the bond linking the substrate to a linker, so here it forms an covalent bond, and we need such kind of a substrate that will form a bond with an linker. A bond must be stable to the reaction conditions used in the synthesis. The means of cleaving the product from linker at the end. Means of cleaving the product from the linker at the end, it means that there should be such kind of an reagents or such kind of an solvents that linker it has been removed or it has been cleaved from an resin bed. Protecting groups or for functional groups not involved in the synthesis. Protecting groups, they are only for an protection, or they are just before an activation, and they should be not been synthesized, they should be not involved in the synthesis of these functional groups. So they should remain silent, or they are not reactive in this synthesis method. So these are the requirements for a solid phase synthesis. Now what is anchor and linker? We will see. The molecular moiety which is covalently attached to a solid support which contains a reactive functional group. So that is said to be anchor or linker. It allows attachment of the first reactant. So that is the role of an anchor or linker, to attach and first substrate, and as soon as the first substrate it get attached, so there will be a chain of an substrate attachment further. The linker must be stable to the reaction conditions in the synthesis but easily cleaved to release the final product. So that is the requirement of an linker, that it should be stable in the reaction conditions and in between it should not cleave. So it should be cleaved at the end and easily it should be cleaved. So that is one of a requirement of an anchor or linker, because at the end we are removing that linker with linker and peptide by separating that linker and peptide, and with that linker resin also or solid support it also removes. Different linkers are available depending on the functional group to be attached and the desired functional group on the product. So what are the requirements of an coupling? So according to that, different linkers are been present, and what kind of an coupling we need to do for a peptide synthesis. So according to that, linkers they are been present in the market. Resins are named to define the linker. So example, Merrifield resin, Wank resin, Rink resin. So in actual, so from this figure you can understand. So here it is a solid support, and this solid support, it is now having an anchor or linker, having an active functional group. So this anchor has a capacity to bind with an solid support, and this active functional group, it should remain free, and this should not attach with a solid support, because this functional group, it will be required for further reactions for a coupling of an amino acid to form a peptide. Corresponding functional group. So corresponding functional group means a functional group here which is green in color, and this corresponding functional group which is orange in color. So that should be suitable one to attach or to form a bond in between, and this is how it happens that peptide it get attached to this functional group, and now there is formation of an peptide or coupling of an peptide until the desired product it has been formed. So these steps they are been repeated. This peptide will again have a functional group somewhere, and it will couple with next amino acid. So we'll see what kind of an resins they are being utilized. So first, that is Wang linker. So every linker has a capacity to bind with different functional groups. So Wang linker, it is suitable for attachment and release of an carboxylic acid. Actually, in case of an amino acid, there are two terminals. So one terminal is that amino acid is having an amino group, and acid functional group, that is carboxylic acid, at one end. So this is a structure of an Wang linker, and after that carboxylic acid group it get couples with its hydroxyl moiety, and now group it has been coupled by using TFA. TFA is one of a solvent, and we can now cleave it. After that, Merrifield linker. So it is suitable for peptide products. So phenyl ring having an methyl chlorine between. This chlorine, it has been removed and it forms an HCl with this hydrogen, and now there is coupling of this carboxyl terminal forming an ester. After that there is deprotection. So this deprotection, it is for removing this Boc. Boc, it is an protecting group, and after removal of this deprotecting group, now amino group, it is active for coupling with next reagent. So now in the next step, again it reacts with an amino acid which has been protected by Boc, and it has been protected by Boc because if it is free, so there will be a chance of an amino acid to react with this oxygen. So that is why it has been protected, and only one site it has been active, and that this site, it is active for an amino acid coupling, and if it comes over here, so it will be repelled. So this chain of an coupling, it continues till our desired peptide, it has been formed. So same step, it has been repeated, and this repetition, it creates a peptide over here. Now it's a time to release this linker from this peptide, and HF, hydrofluoric acid, it has been utilized to remove the linker, and now linker, it is free with a solid support. Now it has been released and desired peptide, it has been formed. After that, next linker, that is ring resin, linker suitable for attachment and release of carboxamide. So here it can be seen that this linker get attached to an acid, and it forms an amide, carboxamide. And again TFA has been used to release this carboxamide. So this linker, it is suitable for attachment and release of an carboxamide. Now we'll see what are the advantages of a solid phase synthesis. Specific reactants can be bound to specific beds. So already we have studied these things, that the specific reactants can only bound to specific beds. Bed can be mixed and reacted in same reaction vessel. Product forms are distinctive from each bed and physically distinct, because the reaction vessel, it is not common for each and every bed. Excess reagent can be used to drive a reaction to completion, because there are limited sites on the bed which are active. Excess reagents and byproducts are easily removed. Reaction intermediates are attached to bed and do not need to be isolated and purified, because we know what kind of reactions a medicinal chemist is performing with the bed and what kind of solutions for coupling are being utilized. So there is no need of an isolation of that intermediates in between. because structures they can be identified once the reaction it has been completed, and individual beds can be separated to isolate individual products. So this is one of an advantage of a solid phase synthesis over other synthesis method, that each bed can be isolated, and due to isolation of an each bed, so individual products can be identified. After that, polymeric support can be regenerated and reused for after the cleaving the product. So whatever linkers and anchors which are being utilized within solid support, they can be again utilized for the further reactions once they are being cleaved after whole process. Next method, that is mix and split method. Mix and split method creates combinatorial libraries where each bed contains one peptide sequence, one bed, one compound. The number of compound increases exponentially with each additional coupling step. So this can be understood by a figure, how it increases exponentially. So what are the process for mix and split method? So the first process is that a bath of resin is divided into equal portion in reaction vessel. After that, each portion is treated with different derivative of first block. After washing, the beds are pulled together in one pot and mixed thoroughly. Then split into equal portion again for coupling to next portion. So this step, this process is continued until a required library has been synthesized. So we can understand this process by a figure. So here it is in figure. These are solid supports having different amino acid for a reaction, and now this amino acid they will couple with the solids. As soon as they are been combined, now they are mixed in one vessel. And once they are mixed in one vessel, why this process it is called mix and split method? Because here we are mixing it, and once it has been mixed, so every reaction vessel is having now similar compounds. But after that, this all reaction vessel, they contains different type of an amino acid which are been reacted. So it means that now glycine and alanine it will form bond, phenylalanine and glycine will form, valine and glycine it will form, serine and glycine it will form, and glycine and glycine it will form. And now with all this compound alanine it will form a bond. So that is why it is said to be a split method. Here we are mixing it, but again we are now splitting it, and due to which we get the products, the desired products. Now here you can see that alanine and glycine has formed a bond. Again, phenylalanine and glycine has formed a bond, valine and glycine has formed a bond, and so on. So this second reagent, it forms a bond or forms a peptide bond with this amino acid which are present in the vessel. So that is why here it splits, and in the first step it mixes. So this process it continues from this process, further process it can be proceeded, because until our library of a desired thing or desired peptide it has been formed, this process can be continued. So here are some examples of an combinatorial synthesis. These are the companies. So Merck, it targeted HIV integrase, and it was having a mechanism action of blocking viral integration. After that, SmithKline become. It was having a target human 5-HT receptor, serotonin receptor, and mechanism it was antagonistic, and for serotonin receptor it was cognitive disorder, After that, Pfizer, the target was farnesyl transferase and its inhibition. After that, Park Davis, KDO8P synthase, and it shows it was responsible for an bacterial action, and due to which formation of an compound, it inhibited an antibacterial activity. It blocked an action. So now we'll see what are the limitations of an combinatorial chemistry. So many times it happens that due to huge amount of an reactions, so there may be formation of an byproduct, or that byproduct which is not of an desired one, so it forms. After that, so many bits are required for combinatorial chemistry, so it does not make it an economical. Probability of finding sample is very less because lot many series they are been utilized and been synthesized, and so there is a very less chance to find a sample. It requires practical details of weight and volume, as it is one of the major limitation of this combinatorial synthesis, because when we are doing a conventional practical, at that time we know the details of weight and volume, but here it is little bit disadvantages. Building and maintaining huge compound libraries is costly business for an industry. So these were some limitations of an combinatorial synthesis. Now we'll see the summary of this session. So the first concept we seen that what was combinatorial chemistry. So it is a concept of producing large number of compounds in a single process, and with a conventional synthesis, a limited number of compounds are being produced, or it is been done one by one. In case of an combinatorial chemistry, a chemist can synthesize multiple compounds in a single processes, and huge number of library it does produce. And mainly this combinatorial chemistry is having an application with a peptide synthesis. So after that we seen advantages of combinatorial chemistry. It is fast because multiple processes are conducted simultaneously. It is economical because the hits can be produced faster and faster, and it's easy. After that, it can be utilized for a drug discovery, and lastly for lead optimization. In case of a lead optimization, whenever lead it is been optimized, there is little bit change in the functional groups or little bit modification in the drug molecule, and with the change in little bit atoms in the drug, so the criteria for lead optimization also been fulfilled. So that is one of a big advantage for lead optimization. After the types, so there are three types: parallel, solid phase synthesis, mix and split method. So in case of a parallel, we seen three classes, that is multiplane, after that T-bag method, and after that spot membrane method. So name itself indicate parallel. In case of a multiplane method, there are 96 wells which are containing an solutions of an amino acids or coupling solutions, and this wells they are dipped, or pins are dipped into a well, and these pins are having an polyacrylic derivatives, which forms an peptide. After that, we have seen T-bag synthesis method and spot membrane synthesis method, in which cellulose paper is being utilized. After that, we have seen the concept of anchor and linker. So it is part of a solid phase synthesis, covalently attached to support and having reactive functional group. And so it is used for coupling with a resin or with a solid support. After that, we have seen some limitations. So some of limitations, like it forms an byproduct, it requires lot many beds, and also creating libraries and maintaining the libraries also increase the cost of this method. Thank you. We have completed module number 7 and lecture number 11, and with this we have finished the medicinal chemistry course. Thank you so much.